This is the chloroplast. As we can see, the chloroplast is an attractive green color. This color is a result of pigment called chlorophyll. As we can see, the chloroplast has two membranes. The green membrane is the outer membrane, while the blue membrane is the inner membrane. Filling the cell is the gelatinous stroma. Now, let's take a closer look at our organelle. These little stacks we see are called granum. Each ring we see is referred to as a thylakoid. The purpose of the thylakoid structure is to create sugars that can be used by the plant. This process is referred to as photosynthesis and occurs inside of the thylakoid membranes. Inside of the thylakoid lie clusters of pigments that are referred to as photosystems 1 and 2, keeping in mind that photosystem 2 actually comes before photosystem 1. They were named in the order that they were discovered, rather than their respective position in the order of the overall process of the light-dependent reaction. One of the aforementioned pigments being chlorophyll. The chlorophyll absorbs photons from the sun as a source of energy for the photosynthetic process. The absorption of the photons by chlorophyll is occurring in photosystem 2. Once this photon is absorbed, the energy will cause photoexcitation of an electron, meaning that the electron will rise up to a higher energy level. As a result of this occurrence, the electron will be passed to a mobile electron carrier, also referred to as an electron acceptor. This leaves photosystem 2 short 1 electron. In order to replace this electron, photosystem 2 splits a water molecule, sourced from the water necessary to make photosynthesis happen. At this point in the reaction, oxygen will be released from the splitting of the water molecule, and hydrogen ions will be released into the thylakoid space, keeping in mind that the latter requires some energy from the electron transfers that occur during the process. Remember this for later. The presence of the conversion of NADPH to NADP+ and of ATP to ADP is required for the reduction step to successfully occur. In the final stage of the Calvin cycle, regeneration, a carbon from one of the 3 carbon molecules is dropped off and ATP is made into ADP. This allows the 2 carbon chain molecules to rejoin and once again become RUBP. This cycle must happen 6 times to produce a glucose molecule, to satisfy glucose's need for 6 carbon molecules. As photosynthesis is dependent on light and carbon dioxide, the abundance of these can impact the rate of photosynthesis. For example, if our chloroplast is left in a dark room for a long amount of time, it will not have enough light to proceed with the process. Therefore, the rate of photosynthesis will decrease. Alternatively, if our chloroplast receives a large amount of sunlight, it will be more productive, and the rate of photosynthesis will increase. And of course, chloroplasts are not invincible. Environmental pressures such as droughts can also impact the rate of photosynthesis, as there is less water to satisfy the need of H2O molecules in light-dependent stages of the process. When exposed to harsh conditions such as acid rain, the chloroplasts will dissolve and unfortunately die. With less chloroplasts, the rate of photosynthesis will decrease. The edge of the jello represents the inner membrane of the mitochondria, while the fruit roll-up on the outside of the model represents the outer membrane. The space between the 2 membranes is the intermembrane space. The blue tic-tacs represent the ribosomes in the matrix, and the matrix is represented with the red gelatin shape. The fruit roll-up on the inside represents the cristae. The chemical energy produced in photosynthesis is stored in the fruit, stem, or roots in plants as sugars. Starch and cellulose are used in cellular respiration. Cellular respiration occurs in the cytoplasm and mitochondria of cells and involves the oxidation of glucose, while carbon and water are released as a waste product. Glucose is broken down progressively, releasing small amounts of energy that is captured and converted into ATP, slowly, in order not to release a dangerous amount of heat energy. Cellular respiration can be divided into 3 steps: glycolysis and the link reaction, the Krebs cycle, and ETC by chemiosmosis. First of all, glycolysis occurs in the cytoplasm and requires glucose, a 6-carbon molecule that is split into 2 3-carbon pyruvates. These molecules are then used in the link reaction that donates hydrogen ions to oxidize NAD+, and then produces acetyl-CoA, a 2-carbon molecule, while releasing carbon dioxide, loss of the 3rd carbon. At the end and beginning of the first process, there are 2 ATP molecules. The acetyl-CoA then leaves the cytoplasm to enter the next step, which is the Krebs cycle, that occurs in the matrix of the mitochondrion. The Krebs cycle progressively strips off high-energy hydrogen pairs from glucose fuel fragments to reduce NAD+ and FAD, and send them to the electron transport system, while 2 carbon dioxide molecules are also being released through diffusion and exhalation. The net ATP at the end of this cycle remains at 2. The third step to aerobic cellular respiration is the electron transport chain, which is driven by protein pumps, or carriers, embedded in the inner mitochondrial membrane and chemiosmosis. The electron transport system is similar to the one in photosynthesis, however, is comprised of 4 complexes in the membrane. When NADH and FADH enter the chain, hydrogen ions are split away, creating an accumulation in the intermembrane space. The potential energy in this space is released due to electrochemical gradient between the intermembrane space and the matrix, which causes the hydrogen ions to shoot through the ATP synthase molecules in the matrix. 2 energy-depleted electrons emerge from the last complex and join with 2 hydrogen ions that were released during ATP synthase. Oxygen then combines with the hydrogen to form water, which is then released as a waste product. Without this oxygen, chemiosmosis and ATP production stop, beginning anaerobic respiration. Fermentation. Aerobic respiration is on average 40% efficient. Meanwhile, anaerobic respiration or fermentation is 2% efficient in producing ATP. Anaerobic respiration occurs in prokaryotes, as well as in plants and fungi, or in the muscles of animals. The reactions produce 2 ATP per molecule of glucose oxidized and are modified forms of glycolysis. In plants, hydrogen pairs are removed from P-gal, reducing it to pyruvate, which produces ethyl alcohol and carbon dioxide. Meanwhile, in animals, the same process occurs, but lactic acid and carbon dioxide are produced, which is the reason for muscle cramping. Factors: glucose and fat availability, presence of oxygen, productivity of photosynthesis in the plants. A very important factor is the amount of hydrogen pairs stripped from glucose during glycolysis in the Krebs cycle. More stripped hydrogen pairs means that there is more NADH available to donate high-energy electrons to the electron transport chain. Due to more electrons passing through the chain, the greater the electrochemical gradient, resulting in a greater amount of ATP produced in chemiosmosis.